Shares of Moderna rising this morning on some comments from Dr. Anthony Fauci. Meg Tirrell joins us with the details. Good morning, Meg.
Good morning, Joe. Dr. Fauci was just on NPR discussing, among other things, the data we saw this week from the Moderna trial of the COVID-19 vaccine. One of the knocks on the way that data came out was that we didn't hear from Dr. Fauci and from NIAID, which are the partners of Moderna on the trial. And of course, remember with remdesivir, it was Dr. Fauci who delivered the news of the results of their trial. So people were scratching their heads about why he hadn't been heard from on the Moderna data. Well, this morning on NPR and also on CNN last night, he said he's cautiously optimistic, having seen the data. He talked about the levels of neutralizing antibodies that were observed at this early stage in the study. They only had 8 patients worth of data or participants to evaluate, and all 8 did show those neutralizing antibodies. He also said that the titers of those antibodies, the level of those antibodies were high, and he did express optimism about that. That was a detail we have not yet seen. He said that the plan is for NIAID to submit those data to a peer-reviewed journal sometime soon, so we should see those data, guys. This is an optimistic update from Dr. Fauci, the person people were waiting really to weigh in on these data. But we do have to remind everyone, this is early days, 8 participants worth of information, still a long way to go to get through this. Back over to you.
And we don't know for sure that even that level or titer of antibodies, whether that generates immunity or not to the COVID virus, Meg. So there. And this, no one has ever said otherwise, that it's a long way off. I just think it helps, actually, as you do, to actually know something about science and the way, molecular genetics and the way that works. Just the ability to take a small segment, as Dr. Gottlieb said, you enclose it in a lipid. It's a small segment of messenger RNA, viral messenger RNA, just one small segment, to put it into a human cell and have it translated into the spike protein that's just like one on a virus, but not the virus itself, and to generate neutralizing antibodies is a positive. And it would help if people just understood a little bit about science to understand that that's a pretty significant step. Whether it eventually results in a vaccine or not is a long way off, obviously. But you know that, Meg. I mean, we were talking yesterday together about using a complete adenovirus, which has a recombinant copy of a viral gene in it. That's double-stranded DNA that you're putting into someone, hoping that that gets expressed so that there's immunity to that. But, I've got my problems with putting another virus in that has a recombinant genotype and everything else. That gets a little, I don't know. This is a much more elegant and presumably a safer way of doing it.
Well, potentially, Joe, and that second approach you just mentioned, of course, is the Oxford-AstraZeneca approach. And so there are pluses and minuses to each one. The drawback of mRNA, of course, is that it just hasn't been taken across the finish line before. So right. There are questions about it. But we'll have to see that in trials.
Right. I don't think adenovirus as a vector that there's an existing vaccine either. Is there?
I think you're right about that. I mean, the AstraZeneca-Oxford approach uses a chimpanzee adenovirus, whereas the Johnson & Johnson one does use an adenovirus as well. But they have tested that substantially in 4 different experimental vaccines across 70,000 people. So we talked with J&J's chief scientific officer yesterday about their approach, and they do have a lot of data on that. But of course, there are still questions. This is a new virus, so we're just going to have to see how all these approaches work.